So thanks for staying this whole day. I tried to keep it short, so because I think every one of you might be hungry again. And I think, although we had a lot of sandwiches, I hope at least, I think every one of you knows the everyday decisions, because we always have to take the decision, what are we going to eat, for example, or what we're going to buy. And I think we heard already a lot about it, that of course our internal signals can help us to decide what we're going to do. And of course, if we are super hungry, we probably make another decision compared to when we are quite sated and feel, okay, like the salad might be good enough for us. And not only the behavior, but also our emotions depend on the processing of internal signals. And again, I want to keep it short. Of course, we have this high cognitive route that we know that we can, of course, make our decision consciously, but there's also this low road that we process our internal signals, mainly driven by vagal afferents. And this interoceptive processing is actually related to a different kind of symptoms. For example, we know that people with eating disorders have problems there, but also it's quite obvious in depression that there might be aberrant interoceptive processing. And especially when it comes to this low road, we can address this via transcutaneous auricular vagus nerve stimulation, as shown here. And again, I want to keep this introduction quite short. We know that we can influence our processing here, but we don't know how far it is impacted by our metabolic state, so whether it makes a difference whether we're hungry or not when we use the stimulation. We also actually don't know much about the stimulation side. So mainly the left ear is used to stimulate the vagus nerve, but actually we can also use the right ear. So that's one question we also wanted to solve. And then in the end, of course, we not only want to have an influence of our interoceptive signals or the processing of it, but we also want to know whether it has an impact on our everyday decisions that we make. So what we did, we investigated the vagus nerve stimulation in healthy participants who came to the lab 4 times. And in each session, we got a 30 minutes baseline measurement of the gastric motility using the EGG. Then we had a 30-minute stimulation phase. And later on, participant received a milkshake as some caloric load. The stimulation went on, and also the EGG measurement went on. And during the whole time, we asked about different ratings, so hunger, thirst, satiety, tiredness, for example. We also asked about the liking of the milkshake. And in the end, participants indicated how much they were willing to pay for food, actually in a real economic decision design. And all participants underwent left and right vagus nerve stimulation in across a randomized order. And it was compared to sham stimulation on the same side as well. Now, with regard to the results, first of all, we found significant site-dependent effects of the vagus nerve stimulation. So compared to the sessions with the sham stimulation, participants liked the milkshake actually more after receiving right-sided vagus nerve stimulation. And in line, they were also willing to pay more for food after right-sided vagus nerve stimulation, whereas for the left side, it was more comparable to the sham stimulation or even reduced, so the liking as well as the bidding behavior of the participants. Now, with regard to the physiological data, as we can see here, we definitely have site-specific effects of the stimulation. First of all, what you can see here is the time frequency plot of our gastric signal, and we can see a clear drop after the milkshake in the frequency, which was followed by an increase in the frequency and especially an increase in power of our signal, which is quite common in the literature. Now, with regard to the power, first of all, we can see that for left-sided stimulation, there was this quite obvious increase in power after the load. However, for the right side stimulation, this was reduced after the vagus nerve stimulation. So in fact here, as you can see, this increase in power after the caloric load was not evident anymore after right-sided stimulation, especially in the end of the phase. It was roughly almost 30 minutes after the load that we can see the biggest difference between the sham stimulation and the vagus nerve stimulation. And actually, it was a significant interaction. Now, with regard to the frequency, we found, again, a significant interaction between the stimulation side and the stimulation condition. So for the left side, we can see that the frequency was higher across all time compared to the sham stimulation, whereas for the right side, the frequency of the stomach signal was decreased compared to the sham stimulation. Again, almost across the whole side, across the whole phases, specifically during the load phase as well. And now, as I said, we were also interested in the in investigating whether this is related to the behavior of our participants, because when we see differences in our gastric activity after vagus nerve stimulation and also in our behavior, of course, we want to bring our data together. So we concentrated, first of all, in this drop of frequency that we could see immediately after the drop. And we had a look whether this was also associated with the hunger rating. So whether participants also had a bigger decrease in hunger, the bigger the decrease in frequency was. So the impact of the load actually on the gastric signal as well. And we found this kind of association that a bigger drop in frequency was associated with a bigger drop in hunger or stronger satiating effects. And this was especially true for the vagus nerve stimulation, but not for the sham stimulation. So it might be that this vagus nerve stimulation indeed helps us to interpret or to process our stomach signals a little bit better. And also during this late phase, when we have the increase in power, but also the increase in frequency again, we found again a significant association of our changes in stomach activity related to behavioral changes. So in this case, the more the frequency increased, again, also the liking ratings of the milkshake were higher. And this was again specifically true for the sessions when participants received vagus nerve stimulation. So here, a change in frequency was associated with greater liking of the milkshake ratings. So to summarize, we had the question, is the stimulation effect of the vagus nerve on our body signals, so on our stomach activity, dependent on the metabolic state? And yes, it was. So specifically after the load, we saw differences between vagus nerve stimulation and sham stimulation, not that much during the stimulation only phase. And most of our effects actually were dependent on the site. So actually the site of the vagus nerve stimulation makes a difference. And just briefly here, actually the results with bigger effects of right-sided vagus nerve stimulation on the behavior, for example, is in line with the current literature. So there are first indications that the vagus nerve has different effects and also different anatomic functions. So we know that the right-sided vagus nerve is also more related to the dopaminergic system. So this might also be more related to the motivation system than in general. In line, we found this increase in liking, but also the increase in the bidding behavior for right-sided vagus nerve stimulation. And then finally, we found support that the vagus nerve stimulation might help to transmit gastric signals to our consciousness, at least, or to the processing of our internal signals, as we found an increased association of changes in gastric motility with subjective metabolic state ratings, as well as increased association of changes in gastric motility with the subjective liking of the milkshake. And with this, we can conclude that there's definitely a potential of the vagus nerve stimulation as a treatment for persons with impaired interoceptive processing, but of course, a lot of more research is needed. And I hope, I made my time. So thanks a lot to all of you. Thanks to Niels Krömer as the supervisor of this work, and also to other people who have worked on this data before already.
Question? Thank you for this. Maybe I didn't follow. I'm sorry for that. But,
it was a rush, yeah.
What is the frequency of stimulation that you use? Target the appearance or apparent fibers of the?
Yeah, so I didn't mention it. I just put it on the slide. So the frequency was a 25 hertz, 30 seconds on, 30 seconds off treatment protocol, which is actually like the standard protocol, which is also used in the treatment, for example, of persons with depression.
Okay.
Yeah, and the intensity was then individually chosen so that it was the below pain threshold, that it was noticeable for the participants, but not painful.
See, mind that it targets more the.
Yes.
She answered already. I want to choose more about the intensity. And just by curiosity, do you try adding intensity to your models to see if it has severe effects with firing?
Not yet. So in the past we did this and it didn't have an effect. But here it's like the first view of the data. You might also notice that there's a difference between the abstract and the data here, because we also found some mistakes. So yeah, this is the first guess that we have from the data. In the models, we also want to include more. Right now, only the session number actually was included, so whether vagus nerve stimulation or sham stimulation was first, so that we can control for the repetition. But we also want to include BMI, age, sex, for example, as the gastric signal, for example, is usually worse the higher the BMI range is. But yeah, most of the results are not affected by this, but I haven't checked it like in a perfect way to be able to present it. Yeah.
All results, I will check. There's also the question, is actually getting to the that is, at least in our study, a very wide variety of, because of the perception.
Yeah, so we know that usually, at least in our data in the past, the perception was more important than the intensity itself, because we also know for sham stimulation, we usually have higher intensities that are needed until the participants notice actually the stimulation at all.
Don't you think that's one super interesting study? And of course, I understand that this is maybe one, and some follow-up might arise. Don't you think that's focusing? Of course, I understand why you do it, but on one high caloric, generally palatable food, right, could be limiting for your conclusions, right? Because you have one type of food, and it's palatable, and it's the same for everybody. So don't you think that's you miss a control, not only a control food, for example, low caloric, but also another control type of reward.
Not palatable or
something. That would be a sort of a baseline. And the second quick question: did you control that effect that you found, the significant effect that you found, especially on the right side of the stimulation? Did you control for likability of that milkshake? Because maybe the effect could be mostly driven by the people who like that milkshake the most.
Which, if you mean the correlation, or?
Yeah. If I understood correctly, like especially when the right side of the vagus nerve was stimulated, then they would like it more and willing to pay more.
Yes, so the willingness to pay, I didn't mention it here, was not for the milkshake, but here we had a range of different food rewards actually. So there we also have this high calories, low fat, sweet or the sugar content as well. So here we have this different kind of range of. So this is just the mean of it, but we also have the analysis then dependent on whether it's high calorie, low fat, for example, and so on. But for the milkshake, so I mean, I think the advantage here is that it's within subject design, right? So they got always the same milkshake and had the liking there. So I don't think that the site then really can explain it, because I think then we would see this effect only in those people who have a high liking and other ones not. So I mean, for the correlation, for example, I also still have to have a look in the side effects and whether it makes a difference between left and right as well. But yeah, as I said, it's within person, so I think at least it's a little bit controlled for this. But yeah, of course, I mean, the gastric signal also depends on what you're eating. So this is also not real food, but a milkshake, right? Yeah. So in long story short, I think, yeah, you could add more rewards and different kind of foods and so on.
Thank you again.